My presentation will be about the regulatory perspective for reliable model-informed drug development. So I will not focus about technical issues or about specific examples. Just I would like to give you some idea about what are the regulatory expectations, or what do we have in mind when we are reviewing or assessing the credibility and the accessibility of submission involving modeling and simulation. So this is my disclaimer. So the view and thoughts here are my own and I don't represent necessarily those of Health Canada. First, I will start with quick definitions, and here I will speak interchangeably about model-informed drug development, about pharmacometrics, and about modeling and simulation approach in general. One of the basic definitions is use of quantitative models to facilitate decision-making process. Actually, in Health Canada, we adopted a similar definition, but we emphasize it about the use of those models to inform and enhance the development and the drug development and regulatory review. This is what I will focus on in the next slide. As you know, the number of submissions involving the use of modeling and simulation have been increased. This is an old review, and you see it is about 65% of submissions to the FDA included labeling or approval based on modeling and simulation. And I think now the number could be higher. And most importantly, I think the impact is higher. Those are few examples about the use of, or the common use of the modeling and simulation in the drug submissions. And you can see that there is very wide range from supporting clinical trials to identifying different covariate to the exposure responses modeling, and we see frequently also investigating of drug-drug interactions and characterization of the pharmacokinetics, particularly for subgroups such as hepatic impairment, and also to support dose recommendation or alternative dose recommendation after approval. And we see also increased use for pediatric claims or submissions, particularly using the extrapolation approach. And also we see also justified use of the modeling and simulation for rare diseases. And also we started to see more submission using modeling and simulation to justify changes in formulations and quality specifications using physiologically based pharmacokinetic modeling and other kind of models. Actually, we already have a lot of applications and expanding use, but I think more can be done, particularly where we have added value or a need for the application of alternative approaches. For example, when we have complex scenario, when we have, for example, what we need to understand the drug interaction between a drug which is metabolized by the liver, and we need to have dose recommendation for hepatic impairment patients because it will be used for those patients. And particularly, for example, if this drug has complicated pharmacokinetic, for example, it is impacted by genotype variations. So in those cases, the use of the modeling and simulation is very useful and is justified. And actually, we can have even more applications in this in similar topics. Also, we can welcome more applications, for example, when the available data or the possibility of obtaining or collecting data is limited for practical or ethical reasons. And this is obviously the case for pediatric patients and the case for rare diseases. So in general, what we think or what usually should be the principle that guides the use of modeling and simulation for drug development. Actually, we should keep in mind that the objective of drug development is to have a better understanding of the benefit and the risk of the drug. So the use of the modeling and simulation should be based, or we should keep in mind that we need to have this better understanding. After that, we can try to have an optimization of the benefit and minimization of the risks. And finally, we can have the usual benefit, which will be a cost-effective development and faster access to the market. But just it is important to have those benefits, but it should not be the focus. It should not be the only thing that we have in mind when we are trying to have our plan in the drug development and to have our plan of the use of the modeling and simulation. So we can achieve this, but while keeping in mind that we need to have a better understanding about the drug and to try to have an optimization of its use. So what are the main considerations in the assessment of the credibility or the acceptability of modeling and simulation approaches? In addition, for sure, to the performance of the model and the quality of analysis, we need to focus on additional things. For example, the model can be developed perfectly and the performance seems to be okay, but we should also keep in mind the following consideration, mainly the quality of data. And I will have a few examples later about this. And also, we should keep in mind the strengths and limitations of the drug, of the model. And finally, we should consider also the feasibility of and the availability of alternative approaches. Actually, the data is very important, and we should keep that in mind that if we don't have the right data, we will not have the outcome that we want to have. This is a general example for here. You can see that in the assessment of the model, we use plots of prediction versus observation. And here you can see in both cases, left and right side, we have the wrong model. But if we have poor data quality or distribution or representation, we will have the wrong conclusions that the model is okay, while it is not. So this is just to say that if we don't have the data that we need to assess the model, this will reduce the reliability of the analysis, even if you do everything right. But I think, first of all, we need to confirm that we have the data that we need. And also, one of the main considerations for regulatory review is the impact of the model. So the model can be fine for a given application, but when its impact is very high or higher, the expectations and the requirements of the development of the verification of the performance of the model and of the documentation, those requirements will be higher. And for sure, those requirements regarding not only the performance of the model, but also justification of the use of the model, or and also the assumptions and the scientific background behind the model development. In the second part of my presentation, I will review with you a few examples about the use of modeling and simulation to support drug submissions. In the first example, I will discuss the use of POP-PK model to evaluate the difference between two formulations. Actually, the two formulations were used in the pivotal clinical trial, and there was no direct comparison between the two formulations. And for technical reasons, this comparison is not feasible at the time of the submission, or so. The sponsor tried to use the modeling and simulation to show that the formation are similar, and so we can have pooling of the data, and we will have higher power. So the question will be, is this use is reliable, and we can trust the model in this application, particularly to simulate bioequivalence study? Actually, the issue with this application was the model was not fit for purpose. For example, it doesn't include the formulation effect, and also it was not validated, or its performance was not tested, so its discriminatory power regarding the bioequivalence was not confirmed. And just if I go back to the first point, if we don't have the formulation effect in the model, that doesn't mean there is no formulation effect. Just that means that the analysis, the covariate analysis or other kind of analysis was not able or was not successful in defining and measuring this effect. So the conclusion actually was this application is not reliable. So in this case, this is an example of where the application and use was not acceptable. The second example will be better. Actually, for just give me sorry, I forgot to share my video, so hello everyone again. So the next example will be about a better use of the modeling and simulation, and it is a case where we have, for example, in the initial submission, we have the definition of the renal impairment subgroups using the creatinine clearance, which is the standard approach. And by consequence, the product monograph dose recommendation for renal impairment subgroups was based on the creatine clearance estimation. However, for clinical reasons or in the development of the clinical practice, the use of EGFR was established to be more useful or more relevant for clinical practice. So there was a need to update the product monograph to include those recommendations based on EGFR. But the problem was the data used to estimate the impact on the exposure from the renal environment was not well distributed based on EGFR. Mainly, you can see if you use the creatine clearance, we have a good distribution of the data, 24 subject or 30 subject, including the normal subject, and we can have a good estimation of the exposure for the normal renal function subgroup, and by consequence for the ratio of the exposure for the other subgroups. While for EGFR, we had just 2 patients in the normal function subgroup. So we cannot trust an estimation of a median or average using just 2 points. However, there was a POP-PK model available, and in 800 subjects. And the question will be, could we use the simulation instead of the observed data? Actually, the model was well developed and included patient with renal function different level of renal function impairment, and its performance was showed to be acceptable for those different subgroups. And you can see that clearly here we have a good distribution between the different subgroups. We have at least 100 subjects instead of 2 for the normal renal function subgroup. So in this case, I think the use of the modeling and simulation even was more reliable than the observed data. And this is an example, because the question was is it better, or the question will be always, could we use the simulation instead of the clinical data? I think this is an example where the use of the modeling and simulation is even better. The second example and the last example will be about the application in the case of a rare disease. And it will be about RUZURGI, and the data and the information presented here are publicly available on the FDA website. And just I will provide my understanding and evaluation of this available data. So RUZURGI was submitted for the treatment of a rare disease. For adult data, we have clinical trials, limited data, but at least we have at least 2 studies, and we have 240 subjects for safety, while for pediatric population, we had very limited safety data, and even we don't have efficacy data. So the use of modeling and simulation, particularly the extrapolation approach, is needed in this case if we need to have an indication for pediatric, and it is based on similarity between adult and pediatric regarding the disease and response to treatment. And the safety data we use it. The safety data is available, as I said, it's limited, but at least there is some safety data. And the model and simulation was used to estimate the exposure in pediatric. But as I said, there was no PKO data in pediatric, and we need to use the adult PK model. For the adult PK model, we have a good data set, for example, 3 studies, and the development model included different covariate, including the weight, which is important if we need to have an estimation for pediatric. However, for the assay was not validated for 2 of those studies, so the FDA reviewer repeated the analysis using the study where we have a validated assay, because we want to have a trustful data. And the development model actually had a good performance across all the age groups, or across all the weight groups. But we need to adopt the model because we need to use it for pediatric population. So the main area where the model can be improved or adopted are the development of the metabolism pathway. But fortunately for this drug, the maturation was expected by the age of 6, and the approval was requested for patients older than 6-year-old. And also we need to take the size or the weight effect, because we need to simulate for pediatric. And the model included already the weight as using allometric scaling. But even if the model in an adult doesn't include that, probably we need to add it to the model. So the model was used to perform simulation, and the covariate effect was not critical. So the model development in one study was okay, because we are simulating a relative exposure, and 2 dosing regimen were simulated. And finally, using the simulation and the limited use in pediatric and based on clinical consideration, one of the 2 simulated dosing regimen was selected. And here you can see a good example where the use of the modeling simulation was very critical and very useful to support the and to support the approval and to have a better understanding and to finally to have informed assessment and decision. So as a conclusion, we can see that for in the assessment of the use of the modeling and simulation approaches in the drug development, we need to verify that the data, the model, and the analysis are fit for purpose, and then to confirm that the conclusion and the claims are within the limitations and the strengths of the model. Actually, the model-informed drug development and the application of the pharmacometric approaches are very important, particularly when we have complex scenarios or complex conditions that should be taken consideration, and when we cannot perform the needed or collect the needed clinical data due to clinical, practical, and ethical reason, as we saw in the last example. And finally, the general conclusion is we can have a better and informed assessment and enhanced drug development using the modeling and simulation approaches in general. And I will be happy to have your questions.
Well, thank you very much for the excellent presentation. If anyone would like to send in questions, please use the chat or Q&A. Well, I'll jump in here. Just out of interest in the case study, you had talked about the pediatric extrapolation scenario. You mentioned allometric scaling, and I just was wondering, were the scaling factors estimated, or did you use the standard fixed exponents?
Actually, here it cannot be estimated approximately because we don't have data in pediatric. So it was actually developed in adults, and I think it was fixed. It was 0.75, which is acceptable if we don't have data. But if we do have data, we can estimate it, but we should also have an assessment to see if the estimation is justified, and if we have physiology or information behind using a different exponent.
Okay, thank you. I don't think we have any other questions at this point, so thank you very much for your presentation.